There we go. I'm recording for Corey. She has to be gone. Like maybe they're actually like playing their game right now or about to. So, and then have an exact recording of this because when I get behind, oh, she would miss some stuff if I just assigned the lecture for recombination stuff today. So we'll pick up with where we were last time. So we had been, and I told you all about where I was when I knew that it was getting real, that this viral outbreak in China might become a big deal. And we've almost gone through 4 years since then, and a lot has happened. And I want to tell you some things about SARS-CoV-2 virus that have not really changed. So the things I have in there about case numbers and so on, those change every year. This does not particularly change. So I wanted to tell you what are the parts of SARS-CoV-2 virus. So it also has an RNA genome, a single-stranded RNA genome. It also undergoes sloppy copying, but not as bad as influenza. So when I first learned about what type of virus was causing the outbreak in China, and I went and started reading about coronaviruses, I thought, oh, good. This has sloppy copy, but not as bad as influenza. So maybe it won't be drastically mutated. Well, wrong. That was wrong. It has mutated quite quick. It does have an envelope. So it is easy to kill on surfaces. Reliably killed off of surfaces with, your standard antimicrobial wipes or chemicals. And it has one particular type of spike protein, which is called S. So its spike protein is designated with letter S. And there are a couple of other ones that are important for what the virus does, which are called N and E. And I might bring those up when we get to immune system lectures, but for right now, we're only going to be concerned with the S spike. So when SARS-CoV-2 interacts with a human cell, it's going to bind its S spike onto a specific receptor that we have called ACE2. Okay, I do have a slide about that, so making sure. That's the specific protein it binds onto, and it enters our cells basically by endocytosis. And then uncoats, and then takes over the cell, and then it packages up, and it leaves through exocytosis. So it does not necessarily kill the cell that infects, but it's definitely going to weaken it at least. So because it's got a membrane, it comes in and leaves with membrane associated processes. So let's talk some about the receptor on humans that it binds to. And I have a visual aid to get from my office. I'll be right back.
Because obviously it's not doing anything.
I have an S spike that my work study, Charles Swigert, who I understand goes by Swiggy around here, 3D printed for me as a part of a class assignment for a different class. And so kind of pass this around to observe, and it looks like this. And this is the S spike that sticks out of the coronavirus membrane. So that interacts with a particular receptor that we have in our bodies called an ACE2 receptor. And these ACE2 receptors can be found in lungs, blood vessels, like basically it's found on the walls of blood vessels. And blood vessels go many places, including the lungs and the heart and so on. And this ACE2 receptor has something to do with blood pressure control. If you take AMP2 with me, you'll learn about ACE protein and what it does. This is a similar one. So it's involved in blood pressure control. It's something that we cannot just get rid of it or change it in ourselves. And it happens to be what this virus binds onto as its receptor to get into human cells. So when people get infected with COVID, because it is entering into many different cell types around the body, because so many places have that receptor, people's symptoms can be really different. And it seems that different COVID strains tend to cause different symptoms. So can you think of a COVID symptom that people used to get and now it seems like they don't when they catch a new infection?
What taste and smell?
The taste and the smell. So that was so weird and so distinctive that in the early days I thought, well, at least there's something to let people know that they might have COVID, even if they don't don't feel anything yet. That symptom is not really showing up with today's strains. My mom likes to text me medical questions that are beyond my knowledge. It's like she's really proud that I teach anatomy and physiology, and she doesn't quite know that it's like really low level. So she'll text me things about medical stuff. And one of them last week was my friend has had COVID and her brain fog is really bad. And like, how long is it going to last? Or she asked me when I had COVID, how long was mine? I was like, I don't remember. It's a long time ago now, but I think 4 weeks. But it wasn't that bad. But I think her friend's is pretty bad, and she's worried about it. So different people presented different ways. Dr. Alderman's dad, who died of COVID, his first symptom was GI stuff, and this was in 2020, I think, in the winter 2020. And some people have had longer lasting heart damage from it, but it's kind of been unpredictable who's going to get what symptom and how long will they last. But there's many places in the body where people can have symptoms. And one of the things that made SARS-CoV-2 or this COVID epidemic so difficult to stop is how it specifically goes through an infection timeline. So this type of graph that we're looking at, if you've got the picture, you can write on here. We're looking at x-axis time, and y-axis is viral load, which is basically like how many viruses are present in the person's body. And for most kinds of viruses, if you build up a large amount of virus in the body, you're going to feel it and notice it and have symptoms earlier on. But with this virus, people could build up quite a large amount before they ever show symptoms and pass the infection to other people before they show symptoms, which that's why it was so contagious and so hard to stop, because you can be walking around feeling okay right now and be breathing COVID viruses on your neighbor and your family and so on. So, that was bad news. That is bad news. When people have influenza, their infectious period lines up better with the symptoms, and both of them are shifted back this way. So by the time you develop a lot of virus in your body, you are feeling bad and you don't feel like going doing things, and you will not spread it as much because you're sick and hopefully staying home to some extent. The other thing that was different and new and bad about SARS-CoV-2 viruses, I think I'm not going to get into this so much, but you all probably remember, like you or somebody in your family taking a test. You're like, okay, I don't have it. And then the next day you're suddenly like really sick and like, oh, I did have it. Test timing and when tests reveal the presence of the virus, that has been hard also to standardize. So our youngest boy, Joseph, he gets COVID more than the rest of us, and he's had it 3 or 4 times. And when he was very little, they were still doing the deep swab. You know, it's going back, hurt so bad, it goes all the way back. And that's what he remembers. Oh, if I have to take a test, it's going to hurt and I don't want to, and so on. So the last time that he had COVID, it was last month, I said, yes, you need to do a test. He was like, I really don't want to. I was like, it's not like that now. It's just on the outside part of your nose now. And then he said, oh, that wasn't so bad. Tests have gotten more accurate and more sensitive. And so the other thing that we all have experienced about SARS-CoV-2 is how many variants there have been. And this is a little bit older of a graphic from probably 2 years ago, realistically, when the general public and me as well was keeping up more with, oh, what strain are we on now? The first one that appeared in China in 2020 is not listed on this list of variants. We would call that the original, and it has a particular number that designates it. That's something like one of these, B.1.1. something. And when the strains were changing so often, each individual one has had one of these crazy long specific numbers, but certain of them also got a Greek letter to designate, oh, we're going to call this one Delta. And I don't know how much attention you all paid to those, but the last one that I paid attention to that had a Greek name, it was Omicron. And there have been more since then, but all of these, whatever their name is and however they're changing, most of the differences are in the S protein. So most of the differences or the mutations have been in the spike protein. I asked Charles, I call him Charles later, I said, what variant is this 3D printed spike from? And he's pretty sure it was basically the original one. Because that was the data set that was available at the time when he made this, which has been at least 2.5 years ago now. It's crazy. So at certain, I'm gonna turn the light off for this. At certain points during the COVID pandemic, there has been a fairly stable period of time where we have this one strain and that's the dominant one, and we say that's the one that everyone has right now. But it can pretty quickly be taken over in a population by a new one. So what this is showing is certainly in fall 2021, when Delta strain had been the most common one, but it was pretty quickly replaced by Omicron strain. And this type of thing tends to happen when the new strain, and this was the case with Omicron. So the new strain that becomes most prevalent, or the most common, this is what makes it more common. So it's better at binding the host cell. Better at binding the host receptor. Or it could be better at getting the host to build more virus. So if it, the average Delta strain caused the host cell to build 70 virus, and the Omicron strain caused the host cell to build 90 virus, mathematically, it's going to outcompete. Or the third thing that can cause one strain to become dominant over another one is that maybe that strain is better at hiding from our immune system. Like, Bryce is so unrecognizable today because it's not even a hoodie. I like it. It's too hot today. Yeah. So, change a little bit about the spike. Immune system doesn't recognize it. So it could be any of these or any combo of these at the same time. And this process, once it happens through sloppy copy, usually, that you end up with new mutants, and most of them are going to be worse at infecting, and most of them are going to be worse at all this stuff. But some, if they're more successful, and the person makes more virus and it infects more people, and it becomes the new replacing variant. And the speed at which this has happened with coronaviruses has been really surprising to scientists. I need to go get my new booster. I am less frantic about it this year because I can see symptoms and deaths and so on declining over time, but I'm still going to get it. This will be the last, literally the last slide in this slide set, but it just kind of shows if you make the S protein represented by this line, and so this is showing how many amino acids are in the whole S protein. So it's got 1,273 individual amino acids to build a spike protein. And it's showing in different variants, where are the places that had a mutation compared to the original. So with the first variant, it had a certain set of mutations at these spots. And with the next variant, it had a certain set of mutations at these spots, and it kind of proceeds through time. But Omicron what in the world? That's so many. So many, which was a good combo to let Omicron be better at all of this stuff, but there is some, another aspect of viruses as they mutate, that is a little bit comforting in all of this. So in general, if we look through human history and about waves of viruses infecting people, they tend to mutate over time to become more infectious. But also less lethal. More infectious, but less lethal. And some of this has to do with the group that they are infecting is building up immunity. And so we will learn in the next unit after this one, how do we build up immunity? And why is it that the first time a new virus comes through our population, we're so bad at fighting it off? Do you all have questions about COVID stuff? One of my questions is why the taste and smell loss and why did it stop? Because that's just, it's really different. And you may think of some later, so. For a long time, one thing that was hard for me when the pandemic first started is that people would come to me with questions like I was an expert. And I kind of was, but I'm not used to having like, people asking me questions that are important and what I say might actually relate to human health. So you all will feel that when you become nurses, I think. Like, people are like, oh, you're a nurse? Well, let me ask you this. You have to be careful not to go beyond your knowledge, but to give them good advice. And like one random thing that will happen when you teach biology or human health things, people are like, I've got this rash, can you look at it? I am not able to help you diagnose your rash. It depends on where it is, I might look. So we're going into a section now that has to do with how do bacteria, this is mostly about bacteria, how do bacteria share genes with each other? And we call this genetic recombination. Genetic recombination. Did you have a quiz? See if I've got the quiz answers at the end. No, they're just totally not here. Sorry about that. I usually put them on the slides. The whole reason why bacteria need this genetic recombination process is because they don't do sexual reproduction, they do asexual reproduction. And I'm making a claim here that most biologists would, is that sexual reproduction has a lot of advantages which go beyond like how it feels or, you know, the opportunity to have offspring. There's other advantages. There's genetic advantages. So what have you learned previously about why, in a biological sense, sexual reproduction is a good thing?
Prevent you from going extinct.
It does prevent you from going extinct.
It keeps
How so?
Because if there's still more of your species, you're not extinct.
Mhm.
What else? Like, genes that you pass on, so like hair color and stuff like that, depending on how you sexually reproduce, you can keep that going, like blonde hair is maxed out. But if blondes would get together, they'd have blonde heads.
Mhm. So it's the diversity of genes allows species to keep going. So for example, imagine that I am a asexually reproducing organism, which means that every offspring I have is exactly like me. I'm pretty sure we would not get along. But imagine, the future generations of me's are going to be exactly like me. And what if the sun gets bigger and brighter, which physicists say will happen, and it starts aiming way more UV rays at the earth and they burn me to death? A silly example, but because I don't have melanin sun protection. And all of my offspring don't have melanin sun protection. So if that's all the genes that we've got, that's all that we've got. We don't have diversity in that aspect to deal with changing environmental conditions. So having sexual reproduction is a way to generate genetic diversity and swapping of genes and crossing over of genes that you learned about in high school biology. For bacteria, if you can't do sexual reproduction and you can't get new genes in that way, maybe you can get genes from other bacteria. And that's what these recombination methods are about. So I want to put here some things that bacteria don't do. That they don't have because they don't do sexual reproduction. They don't have 2 copies of each chromosome. They only have 1 copy of every gene. They don't have dominant and recessive. How nice. How nice if you're studying the genetics of bacteria and like, well, you've got the gene or you don't have the gene. So there's no dominant and recessive. There's no Punnett square. It's like, whatever you've got, so does your offspring. It is literally the same. So instead of dominant recessive, they either have the gene or they don't have the gene. It's much more simple, yes or no. Can you do X with this gene or can you not? Genetic recombination is a way for bacteria to get new genes, maybe, and give some genetic diversity to their offspring, which gives their offspring a better chance to survive. So there's 3 big mechanisms of this. And I really like these overview cartoons that your book has. And it's describing with some helpful cartoons what are the 3 mechanisms. So we're going to list them on here and then we'll go through what are the details of each one. So the first one is called transformation. That goes with this cartoon on the left. There's transformation. There is a version called transduction. In the middle. And then there is a version called conjugation. There was a test in here right before, did you see? Dr. Pickard will sit in the chair, sometimes fall asleep. He does not do that in the chemistry majors class. I'll ask him why. I don't know why. But in the non-majors class, he will sit here and fall asleep. Sometimes. So in all of these, there is bacterial DNA moving between bacterial cells. So that's the same for all of these. So in some way, bacterial DNA is going to move from one cell to another. That's true for all of these. What's also true for all of these is that the DNA may or may not be useful. There is not a choice of DNA to donate. It's just kind of by chance, so to speak. By chance. And then the third thing that's true for all of these is if the DNA is going to be inherited, and be passed on to the offspring of the bacteria, it has to be integrated or joined with the host DNA. Or we'll call it the DNA of a new cell. So see how in here's some DNA and it gets into the cell. And then down here, it's showing that it's joined with the DNA of the new cell. Same in this case, transduction, and we've got, this is a phage that's doing this. We've seen this before. Here comes the DNA, and it can get into this cell, but it has to be joined with the host, or the new cell's DNA. Let's add on to this one. Also the word stabilized. Stabilized. Because in our last example of conjugation, the DNA is moving as a plasmid. And it becomes stabilized in the new cell as a new plasmid during conjugation. So I'm going to use, I like to use an example when I teach this that, like I am going to share some information with you that came from my house, and it's something on a piece of paper. So I'm comparing a genome to all the papers that exist in a house. So that's my scenario that we'll talk through. Okay, I want to first start with transduction, actually, because this one is most familiar to what we've been talking about recently. So if you'll skip forward to transformation. So transduction, did I just say transformation?
Yeah.
I always tell students, don't mix these up. Transduction. And here's how I remember this one. So did you ever play duck, duck, goose when you were little? Okay. So there is a game in the US that's called Duck, Duck, Goose. And everyone sits down in a circle, and someone stands up and they go around and they tap everyone on the head and they say, duck, 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 duck. And when they say goose, you have to get up and you chase them and you try to touch them, and then if you touch them, then what happens?
Then they go in the pot.
They go in the middle, and you keep playing this game. So, so the way I remember this transduction is like a virus phage ducks down, like squats down and delivers the DNA. That's how I remember this. It ducks down. And that's not even related to Duck Duck Goose. I don't know why I told that story, but anyway. So in transduction, a virus is moving DNA between cells. And what happens is when the virus packs up, like this picture, let's see if I got it. Okay. Do not love this picture. It's actually not the right picture that I wanted. But when the virus assembles, the virus is going to accidentally include host DNA instead of virus DNA. There is both, every virus that packs up to leave a host cell, it's supposed to be getting some DNA to take with it. And it's supposed to be DNA from the virus. It is possible that the virus accidentally gets some DNA from the host cell, which is like this right here, and it packs up host cell DNA, and then it delivers bacterial DNA to a new bacterial cell. So this picture is showing this concept that the green DNA is actually from the virus and the pink is from the bacterial cell. And what we need to do to make this picture make sense is to draw onto this virus right there, a little piece of the host DNA also. That's what's missing. That's why I don't like this picture so much. So this virus, once it leaves this cell, it'll go and infect a new cell. And now that little piece represents new bacterial DNA for this new host cell. It got accidentally carried along when the virus packed up to leave. So here's a dumb example. Our boys now are old enough to kind of pack their own clothes for a trip. But if you ask a very little kid to pack clothes for a trip, and they're like, here's a suitcase, fill this up with clothes so we can go on a trip. Like you would think that they would get their clothes, but what if they just filled it up with some clothes from the house? I got clothes, I got a suitcase full of clothes. That's like a virus, when it's packing DNA, it's not checking what DNA that is. It's like, yeah, I got enough DNA. I'm ready to go. Let's leave. And it didn't get the right DNA. It got some DNA that didn't even belong to it. And then it brings that to the new cell. So if you were this cell and you could think and observe and you're like, oh no, here comes a virus about to infect me. This virus is lousy at actually infecting you because it doesn't have the right genes. So that's good. this cell's not gonna die from a viral infection because this virus is messed up and doesn't have the right genes. And it gets something new. It could be good, useful DNA. It could be DNA that the cell already has. It could be something totally pointless. So when a new cell gets DNA, it's like if you came to my house and you got the chance to get 3 papers from my house of all the papers that exist. I can tell you right now, you're probably leaving with some kind of garbage. I mean, it could be Subway coupons. It could be instructions for an appliance that we don't even have anymore. It could be a PALS receipt. What if you got my Social Security card? That would be useful if you're a mean person. Or what if you got, these don't exist on paper at my house, but like, what if you got a copy of test 3? Ooh, value. What if you got the list of the unknowns and the key for something? Ooh, value. But the large majority of random papers that you could get from my house would not help you at all. Total garbage for you. Yeah. What if you got 1 page of a sermon from 3 years ago that could help you? So many papers in my house. So in this case, that's transduction. So I want to go back to the overview page. Where's our overview page? This. And write some things about transduction to sort of summarize it. So transduction, there are restrictions on the length of DNA. The DNA that's moving has to fit inside the new virus, so it can't be an endlessly long piece of DNA. And restriction can only be, it can only move DNA between related species. And then just a reminder, this one has to have a virus. And imagine it, there goes, it ducks down, delivers DNA, transduction. This virus has to be able to infect, the new cell that infects has to have the same receptor as the first cell. So they can't be drastically different kinds of bacteria, like it could move between 2 E. coli. Or maybe between 2 Staphylococcus epidermidis, but it's not going to move between totally distant related bacteria. That's transduction. The next one that we're going to look at is transformation, which is this one. And we have several more slides about transformation. And there's a really famous and important experiment that demonstrated transformation, and it was by a gentleman named Griffith. Griffith was studying bacterial infections in mice. And the reason he was doing this is because this same bacteria can infect people. And it's called Streptococcus pneumoniae. We're going to use a weak version of this next week in lab, strep pneumoniae. And strep pneumoniae comes in 2 different forms. It comes in a form that we call rough, which has wrinkly, bumpy looking colonies on a plate. And it comes in a version that we call smooth, which has a shiny, round colony on a plate. And Griffith, during his experiments with the mice and these bacteria, he determined that if you infected mice with the rough strain. And it was not just this one mouse, it was groups of mouse, groups of mice, you know, for statistical power. So, he was literally injecting them into the mice, I call them mice. And he saw that if you put the rough strain into the mice, it would live. It's not lethal. So that was his first, or one of his first sets of experiments. If he did the same thing with smooth strain in groups of mice, he would grow some smooth strain of strep pneumoniae, inject it into mice. They would all die within a certain number of days, which is really sad. Medical research, there you go. So they would die. So there's something about the smooth strain that makes it more deadly than the rough strain. Then he did something more interesting and different. So his question was this. Really is what he's trying to figure out. So he wants to know what makes the S strain deadly. And he had two ideas about what this might be. So either maybe it's the action of living cells, or maybe it's some type of poison or toxin that the cells make. Two options. It's something that living cells are doing to the mouse, or it's something that the cells are making as a poison and sending into the mouse. Two options. So he decided to boil broth culture of strep pneumoniae, and he used, Bunsen burner like we have. Boil the culture, get it really hot, kill the S strain. And we call that heat killed. He killed the S strain and he injected that into mice, and he found that they lived. So that makes him think, okay, it's not a poison, it's something in the living cells that's making them deadly. How's this seem so far? Did any of you learn this in high school? You've probably heard of it, maybe. Yeah. Super famous experiment. And then Griffith did something that I would never think to do this. And this is why I'm not a research scientist. I would rather teach. He decided to mix. He decided to mix two strains together, which individually are not deadly. So the rough strain living, he knows that's not deadly. The S strain, heat killed, he knows that that's not deadly. He mixed them together and injected the mixture into the mice, and they died. Pretty much just like they would have died if the S strain had been living. And when he checked the blood of the mice, he was able to isolate and grow on an agar plate living S and living R, both. How strange. How strange. And at first he didn't believe that he did it right. So he kept on doing it, and more mice sacrificed for our knowledge. And he kept on getting this result. And then the question is, like, how do these Where are these S coming from? Is the R bringing them back to life? What is happening? And through some careful research and a lot more people working on this with him, he eventually figured out what was happening. So let's go backward to this slide, and your picture here is telling you what is special about S is that they have a capsule. S has a capsule. R does not have a capsule. So question, think back to unit 1. What's the capsule made out of? It is glycocalyx. Remember that? That's what it's made out of. Glyco. Sugar. Yeah. It's made out of sugar. Smooth has a capsule. Rough does not have a capsule. Smooth has a capsule. Because it has genes to build capsule. And I don't remember if it's 1 gene or 3 genes, but it's not a big piece of DNA that lets it do that. What if you came to my house and you happened to find a paper that let you build a shielding cloak around your house? That'd be awesome. That does not exist at my house. I don't know how to do that, but that's what Smooth has that Rough does not. It has a little stretch of DNA that tells it how to build a capsule. So killing the S strain and putting this into the mouse, there's no live bacteria to kill the mouse. Why does that matter? So what the capsule is doing, if you have a capsule, you hide from the immune system. And if you have a capsule and you hide from the immune system, the bacteria are going to multiply in secret inside the mouse because our immune system can't tell that the bacteria are there. The capsule is like a invisibility cloak. If you got an invisibility cloak, you can multiply, and all your offspring will have invisibility cloaks, and you build up and you build up and you build up until you kill the mouse. And that's why the S is deadly. It's deadly because it's got hiding ability. It hides because it's got capsule ability, and it's got capsule ability because it has capsule genes. Griffith was not, like, people didn't know about genes back then. There was no DNA structure. They didn't know any of that. So this was the start of figuring out what is a gene and what is the heritable material. So what was happening when Griffith mixed the two? These were heat killed. And when they were boiling, when these were boiling, they leaked DNA out into the environment. They're dead. Be like if you blew up my house, but here come all these papers flying out of the house. There they are, papers everywhere. Some of that DNA could be picked up by R strain cells. Our strain cells, we know now through experimentation and a lot of different proofs, the bacterial cells can pick up random DNA. Pick up random DNA pieces, most of which would not be useful. But the ones that picked up capsule DNA can now build a capsule and can now hide from the mouse immune system and multiply in secret and kill the mouse. That's what happened. They picked up DNA. All of the rough strain cells that picked up other kinds of DNA wouldn't have been helpful. The mouse immune system would have, maybe killed them or maybe not, but the capsule DNA was the key helpful thing. And we would say that that transformed the rough strain into the smooth strain. So that's where the term transformation comes from. Smooth cells getting a new ability. On your exam, which is next, is it Wednesday? We moved it today. Yeah, next Wednesday. One of the questions on the exam is to list the types of bacterial recombination. Like list the 3 types, and I know we haven't talked about the last one, but that'll be pretty short on Friday. List them and spell them correctly, and that's a 6-point thing. And then there are questions after. Like, okay, you got the list, and then here's a question, which one of these, blah, blah, blah. And there's some questions in amongst that about this Griffiths experiment. So that's why there's multiple slides. That's why I wanted to go to that one today and make sure that we finish that one today. So let's end. We'll see if we've got time to put our summary notes on here. Okay, so transformation can be between widely different bacterial species. They don't have to be as related to each other. They just have to be able to pick up some outside DNA and get it inside the cell. And a lot of bacteria have the ability to do that. So this lets bacteria share information, even if they're not related to each other very well. It can also transfer bigger pieces of DNA. And because there's not a lot of restrictions on it of who can transfer to who and how big the DNA piece has to be and stuff like that, this one is the most common. This one is the most common of the 3 types. And it also has the coolest experiment, Griffiths experiment, to describe it. So on Friday, we will talk about conjugation. I think maybe there's 2 slides about, don't even have us, there we go, 1 slide about conjugation, and then we'll review. So we'll do test review. The test will be Wednesday, but Monday there'll be just some new material that's be on a later test. That's the plan.
So do we need to do the quiz that's due on Wednesday for learning?
I need to move it. Let me think about that. I gotta move I'm gonna move it And you should plan on doing that quiz on Monday. Yes. I gotta change that in Canvas. See you.
Okay. Yeah.
You better than this.
I think you will.
Yeah. There you go.
I don't know.
See you.
You too.